Hi everyone. Thank you so much for being with us today for our first FCA webinar of the year. We're joined by the team at Young Henrys and the University of Technology, Sydney. So thank you, Richard, Alex, and Kira for joining us to discuss turning brewery waste into climate wins, cutting livestock methane. Um, It'll be a really great session, and we will hear from all the speakers first and then move to a Q&A afterwards. But just to get us started really quick, FCA would like to acknowledge the traditional custodians on the land on which we're meeting, and pay respects to elders past and present, and extend that respect to any Aboriginal and Torres Strait Islander people who may be present today. Noting that we're coming to this webinar from all across Australia. I'm coming to you from Melbourne on the lands of the Wurundjeri people. Our first speaker today is Richard Adamson. Richard is a non-executive director and founder of Young Henrys. He founded Barron Brewing in 2005 and created the Black Wattle Ale, which started the trend of using Australian native ingredients in beer. Richard went on to start Young Henrys in 2012. Young Henrys is a brewery and distillery in the heart of creative Newtown in Sydney. Its approach has been to be at one with its community, and it has expressed this by embracing music, art, and culture. And you may have seen some of their collaborations where they make unique brews with the likes of the Foo Fighters, FBI Radio, Kylie Kwong, and Art Gallery of New South Wales. Richard was the president of the Newtown Precinct Business Association, and a member of for 4 years, which launched the first co-working space founded by a local chamber of commerce in Australia. Richard was a founding member and vice president of the Inner West Brewers Association and is currently a director of the Independent Brewers Association. Richard is committed to ensuring a healthy brewing industry and has contributed to this through the development of the Certification 3 in Microbrewing course, which is delivered through TAFE New South Wales. And he continues to foster the development of new entrants to the industry as a part-time teacher there. So thank you so much, Richard, for joining us. I'll hand over to you to discuss a little bit more about what Young Henrys is doing in this space and their project to turn brewery waste into a methane inhibitor. Thanks, Richard.
About talk about partnerships with UTS and how UTS work collaboratively with businesses to try and solve big problems like climate change. So yeah, beer positive is what we're calling this project. But yeah, a little bit of background on Young Henrys in terms of our environmental sustainability goals. So we're a certified B Corp. For those who don't know what that means, it is a certification program. You need to be audited, and it's audited based on a whole bunch of criteria: governance, sustainability, in terms of environment as well, labour, engagement with the community, all those types of things. We're also a signatory to the UN Global Compact, which kind of informs a lot of the decision making that we have in regards to this space. And obviously, we're in partnership with UTS and the Green Biotech Hub. So a little bit about kind of what shapes our thinking is the UN Sustainable Development Goals. So these are, as you can see, very lofty goals in terms of no poverty, no hunger, life below water, climate action, etc. And it can be a bit daunting when you look at these as a business or as a farmer, I guess, as well, about, well, how can I impact these on a micro level on just my day-to-day business, and what can I do to be part of this? The UN kind of distill those 17 down with the UN Global Compact, and this is talking about corporate sustainability. It starts with the company's value system, principles-based approach to doing business, and taking responsibility for those areas of human rights, labour, environment, anti-corruption, and it's good practice to not to harm one another essentially, and try and do the best you can as a business as well. So that gets distilled down to these 10 principles, and you can see that there's some broad chapters around human rights, labour standards, environment, and anti-corruption. So we as a business have looked at a lot of these principles. So labour standards is one. We take our responsibility to corporate citizen very seriously. Obviously, we were selling alcohol, so that comes along with a social license as well. So we need to be conscious of how we market our products. Look, steering clear of being sexually suggestive and all those types of things. I guess that one of the bigger problems the brewing industry faces is it's been exclusively a male-dominated industry, and even marketed directly towards men to the exclusion of women, which I think has been to the detriment of the product in the long run. So, I think equality and equity is something that we need to be focused on, and look at the gender pay gap, and look at the representation of women and minorities within the brewing industry as well. And that's something that we've been focused on. But in terms of narrowing the scope for this particular talk, it's probably worth talking about environmental sustainability in a bit more detail. And there's a lot of challenges in the brewing industry in terms of trying to be as environmentally sustainable as possible. Energy, we are a big user of energy. We are right in the middle of agricultural supply chain, so we need to be conscious of the production of ingredients, transportation of ingredients, efficient use of those ingredients, and how we dispose of the byproducts that come from our processing of those ingredients as well. Obviously, packaging, a big part of producing beer, and quite a large carbon footprint as well. So production, disposal, transport, use of water. So we are a big use of water, and that's one of those UN sustainability goals as well, is life below water, and also water consumption. And then we produce CO2 through the production of our products as well. So a couple of things that we have done. We were very fortunate to partner with Pingala, which is a community solar farm. So those solar panels on the roof of Young Henry's are owned by the community and leased by Young Henry's. So that generates up to 45% of our on-site requirements for electricity. Those people that invested in those solar panels get a 6% return. And we also source a amount of our electricity from a solar farm via the grid. So we've got a PPA in place for that. We've looked at our sort of grain and water usage, and we've installed a high efficiency brew house, which is kind of a big brewery technology on a small scale. That lessens the environmental impact, but also, really in the long run, it helps our bottom line as well by being more efficient. So that's definitely a win-win. In terms of byproducts from our processing, look, spent grain is a large one, and like most breweries around the country, it does go out to cattle feed. And we've had feedback from our farmers that come and pick up the spent grain, that it really helped them through periods of drought. The last severe drought, I know that was the one farmer said that he was able to sustain his herd well beyond what he would be able to do if he didn't get the spent grain. So we're really pleased that we can have that support, and that's a mutual benefit to both of us, because otherwise we'd have to dispose of that spent grain, and it's great that it goes out to a high value cattle feed as well, in terms of its nutrient source of protein particularly. And that kind of leads us to where we're going. So knowing that we have almost like a grassroots supply chain to farmers, we're obviously a user of agricultural products. There's more agricultural land set aside to growing ingredients for beer than there is for wine by a big margin. We are a net exporter of malted barley. and our hop industry has grown pretty dramatically as well. We've moved from it being a commodity-based product all about alpha acid into one which is more now about flavor and impact in beer, which is exciting, and those hops are sought after around the world. But looking at this large bulk of waste product and going into already going out to farms, it kind of sparked me into thinking of how we could do something maybe a little bit more interesting with it. Um, the other thing to consider, obviously, is our production of carbon dioxide. So on a real basic level, we're taking the malted barley, which has already been pre-processed to a degree and makes the enzymes in that barley seed available to us to convert starch to sugar. And then the magic of yeast creates alcohol and CO2 from that sugar. Alcohol obviously being the product that we want to be in the beer. And CO2, we are a user of CO2, but we're emitter of CO2 too. So we definitely create more CO2 than we would use within the brewing process. And there's some interesting kind of conundrums around CO2 as well that are worth talking about. So, in our conversations with UTS and the climate change cluster, we did look at what they were doing in terms of trying to tackle these big problems of climate change and what industry could do. And it kind of poses the question, what if drinking beer did something positive to the planet? So what could we do within our operations to do something positive? And then the other question is, can we transform two waste streams into a revenue producing product? And I think we need to be very conscious of the fact that any sort of thing that we do to mitigate climate change and mitigate our emissions needs to have an economic imperative as well. Otherwise, it's just really not going to get off the ground. And making sure that that's the case, we can get those flow-on benefits as well. So why are we interested in this? I think, like Scope 3 emission reporting is becoming mandatory for reporting industries. So that's in place now, as I understand it. We're also facing one of the biggest challenges and opportunities in our lifetime. So, with moving to a low carbon economy means that, things will have to change, but there is opportunity and business opportunities and opportunities for us as a country to export to other nations and really grasp that. And as I mentioned before, brewers are in the middle of an agricultural supply chain. So, as I said, we're end user for a lot of the agricultural products grown within Australia. We produce a product that is really an agricultural-based product, and then we have waste streams that also go back to the farm. So we're kind of already in play in terms of the circularity. I mentioned some CO2 contradictions before. So we're producing a lot of CO2 with fermentation, and in most cases in brewing, that's just going out. That's just a fugitive emission that goes out to waste. CO2 supply has been at risk and has been pretty volatile in Australia coming out of COVID, and pricing's all over the place. And you get this real unusual contradiction where we're all hearing there's too much CO2 in atmosphere, but we don't have enough compressed food grade CO2 available commercially. So that seems kind of crazy from any way you look at it. And as I said, it's farm to glass to farm. So malted barley makes beer, spent grain is resulting waste product, and that's going out to cattle feed. Just a quick one on emissions and brewing. So annual emissions from one of the largest breweries is 4.1 million tons. Of our emissions, 85 to 90% Scope 3, and there's very few breweries that are actually capturing their CO2 and reusing. Scope 3 from cattle farming, you've probably seen these numbers and variations of these numbers, but we do know that methane is a big issue that needs to be tackled within the industry. And in some ways, we need to recognise that red meat has as an image problem. The numbers of vegetarians in Australia are growing. 50% of the 18 to 24 age bracket cite the environment as the reasons that they are vegetarian. That's worth considering. And there is the growth of plant-based products. I don't believe that plant-based proteins will ever replace red meat. I think the requirement for protein was always going to outstrip what the Australian red meat industry can produce. And we're probably talking about red meat being a high value product that is probably eaten less frequently. Just something to recognise, I think. So what if breweries could inset reduction of emissions and impact scope 3 of cattle farmers downstream? That's kind of the big question that we're kind of tackling. And how would we do this? It would be beer through beer positive, and it's about CO2 self-sufficiencies for breweries, creating a natural product from waste, and reducing agricultural methane emissions. And this is our target of 50%. We'd love to say we're doing that right now, but we are working towards it. So how a quick look at it before, I don't want to get into Kira's territory too much, but it's about installing algae bioreactors within breweries alongside carbon capture systems. So we capture that CO2 from the operations, we grow algae, and then we use that algae to supercharge spent grain. So I guess the inspiration of that was looking at the work with red asparagopsis, the macroalgae, and then asking the question, can microalgae do a similar job? There's some massive benefits for using microalgae. We can put that onshore. We can co-locate that in CO2 emitting industries where we can use that CO2 to accelerate the growth of the microalgae. And then in the case of breweries, we've already got spent grain going to the farm. So it's really about piggybacking onto that built-in supply chain that is there already. Which leads us to the great statement I never thought I'd be able to say, which is drink beer, eat steak, save the planet, and that's what we're all about. So that's a little overview, and then there's much more detail coming.
Great, thanks, Richard. I think, yeah, some great insights there. I think, yeah, what if drinking beer did something positive for the planet is a really great catchphrase, but also looking at, you know, your discussion around scope 3, supply chain risk, pricing, things like that, can see how this all ties together and benefits agriculture. So looking forward to learning a lot more about the detail from Kira and Alex. So as I said before, we are joined by two academics from University of Technology, Sydney, who are going to discuss some of the technicalities and the work and how we bring about sort of this microalgae and turning brewery waste into a methane inhibitor and reducer. So I'll introduce Alex and Kira. So Dr. Alex Thompson is the Industry Engagement Manager for Climate Change Cluster Research Institute at the University of Technology, Sydney, where she leads the engagement of research in creating climate solutions for the planet, from the planet. With a PhD in marine ecology, she was formerly a lecturer in the School of Life Sciences at UTS and a Science and Technology Australia superstar of STEM. Through the Deep Green Biotech Hub, she spearheaded the world's first dedicated algae accelerator program, joining biotechnology and entrepreneurship, creating opportunities for STEMpreneurs across New South Wales to accelerate algae innovation and engage with science. She is the co-producer of the Splendor in the Grass Science Tent, a TEDx speaker and a big fan of algae and how it can change our planet. And she's also joined by Kira Picknell. Kira is a third-year PhD candidate at the Climate Change Cluster at the University of Technology, Sydney, pursuing an industry PhD. Her research focuses on using brewery waste and microalgae to create sustainable cattle and sheep feed, aiming to reduce methane emissions and enhance productivity. She's graduated with a Bachelor of Animal and Veterinary Bioscience from University of Sydney in June 2020. Two, driven by a passion for sustainable industry practices and innovation waste utilization in agriculture. Kira is a 2024 UTS 3-minute thesis finalist, winner of the 2024 Communicate Your Research Award and a BeefX at BeefX, and placed third in the 2024 New South Wales Falling Walls Lab competition. So thank you for being with us, Alex and Kira. I'll hand over to Alex to talk first, and then we'll follow by Kira. And then after their two presentations, we'll have a Q&A section. So feel free, everyone attending, to put your questions in the chat or the Q&A function on Zoom. Thanks, Alex.
Thanks, Paul. I'm just checking that my screen is up. Yeah, great, fantastic. So as Paul mentioned, Kira and I are from the Climate Change Cluster Research Institute at the University of Technology in Sydney. I just want to give a bit of a brief overview of what we do as a research institute and the types of projects that we work on, and then a little bit of a history of this collaboration with Young Henrys, because it has been going for a number of years. And I think it's always that interesting thing with research projects is when you kind of hear about things in the media or things kind of pop up about it, people think it's been something that's just sort of happened very quickly. But this project has been going for, Rich, like 7 or 8 years now, I think we're at. So there's been, yeah, it's been a long time. And I think one of the things that this project has done really well, and we hope to kind of continue doing well, is really consulting with a lot of the stakeholders that we work with, and that we hope this solution will end up working for. So I just want to talk a little bit about that, and then Kira is really going to get into the nuts and bolts of the project that we've had underway for the last couple of years. So the Climate Change Cluster is a research institute. It sits within the Faculty of Science. We have a focus on, particularly in the marine space, around marine research, but it's really kind of underpinned by looking at climate mitigation and climate adaptation. So a lot of the research we do is really focused on big industry problems, and how can we implement some of these solutions that either help to look at how those industries can adapt to climate change or mitigate climate change in the types of products and services that they offer. We obviously really focused on this as a challenge. What started off as a research institute that was pretty focused on sort of our marine environment has then kind of expanded to say, how do some of these things that we're researching help to actually impact and change our planet for the better? And obviously we all know the challenges of climate. I'm not really going to go through that. I feel like everyone here is pretty well versed in that. But how we kind of address the problem is really what we try and do as a research institute. So the Research Institute is pretty big. We've got 4 main teams. There's a team that looks at coral restoration. There's a team that looks at ocean microbes and water quality, one that is an oceanography team. And then we have one specific team, which I'll talk about today, which is the algae biosystems and biotechnology team. Before I kind of dive into how we kind of do this type of work, I want to kind of talk a little bit about what algae actually is. So we work with across all algae, but algae can be broken into 2 main groups, the microalgae and then the macroalgae. The macroalgae is the things like the seaweeds and the kelps, and microalgae is the tiny single-celled organisms that you get in ponds and dams and can cause algae blooms. It's a really, really diverse organism, and the project that we're focusing on today is specifically to do with microalgae. So microalgae, there's anywhere between, they say, 70,000 to 1 million different species across the planet. It's full of things like proteins and lipids and carbohydrates. It's got lots of little micronutrients in it. It's a really kind of useful tool for scientists to use in things like biotechnology, because we can find an algae strain that has specific nutrients or qualities or grows in a particular way that may be applicable to different types of industries. So there's lots of different algae that we work with in our lab. I think at any one time we have about 200 that we're working with. And really, if we have a problem that we're trying to solve, we can usually try and screen for an algae that might help to address or solve that problem. The really great thing as well about microalgae is it grows really, really fast, and it means that we're able to kind of work with it pretty quickly as well. So this algae team that we kind of works with algae in our lab is a pretty diverse team. There's everything from engineers to robotic experts to people that work specifically with marine algae or freshwater algae, all the way through to people that work with things like breeding protocols for kelps. So really, we try and tackle problems with a really, really diverse team and try and come up with solutions that are pretty multidisciplinary. At any one time, we could be working on a project that may focus on how we can grow a different type of algae using artificial intelligence, all the way through to making a new type of brick or tile out of seaweed waste. So we try and work across different supply chains as well. So people who may be growing algae may have an excess of it, and we try and help them to kind of come up with solutions for using that, all the way through to people like with Young Henrys, where we're trying to use algae as a solution for capturing carbon and then turn that into a useful product. We do a bunch of different projects, and some of these you might have seen. Obviously, the Young Henrys one there is listed, but we also work with people in the fashion space, so generating new types of fiber as an alternative to synthetic fibers. You can kind of use things like seaweeds to make alternatives to those, all the way through to coming up with pigments for food. So we've done a number of different projects with industry partners where they've come with a diversity of different problems, and then trying to find an algae or a solution with algae is kind of what we specialize in. I just want to show a little bit of a snapshot, because I think sometimes it can sound pretty amorphous if you're kind of talking about the different types of solutions you can come up with algae. But we use algae for a bunch of different things, all the way from fertilizers to food additives. Specifically in our lab, those photos on the right there are some different products that we've developed using either seaweed or microalgae. We've developed everything from plastics to, as I said, fiber and fabric, bricks, tiles, plasters, as well as a number of food additives, nutraceuticals from algae. It's always nice to see photos rather than just kind of sounding like people are kind of just saying there's a bunch of things out there. With this project, this, as I mentioned, has been going for a number of years. And I just kind of want to talk through a little bit how this project kind of began and where it's kind of gone in the years that we've kind of been running it. So I think Rich and our research director had a chance meeting somewhere. Am I telling the story right, Rich? And decided that maybe there was something in this whole relationship between yeast, beer, and algae. As Rich kind of alluded to, obviously with beer fermentation, you get production of CO2. Algae does the complete opposite, where you're, growing algae that is capturing carbon dioxide and emitting oxygen as a result. So you've got a really nice kind of complementary reaction happening there. And I think there were some conversations around what could we actually do with this. So that kind of kicked off a couple of projects back in 2019 and 2020, where they were co-funded by the federal government through a scheme that's no longer around anymore, Innovation Connections, where we really looked at, first of all, can algae actually survive in the brewery? Is that even going to be a thing that's going to be viable? I think there were a number of different algae that were trialed down at the Newtown Brewery to sort of see if they could sort of survive, which ones would sort of thrive in that slightly higher CO2 environment. And then once we had a kind of number of those microalgae strains that were tested, then it was kind of the big infrastructure task. How do we actually kind of refine this process? How do we sort of maybe capture some of that CO2 that's coming off the fermentation vats and feed it directly to the algae in a more targeted way so we can actually track the CO2 that we're capturing? We implemented some equipment in the brewery that helped us to do that. And if you go down to the Newtown Brewery today, there's still, and you kind of see the picture on the left there, those big, what we call them, cage or bag reactors that are full of algae that are utilizing some of the CO2 that's on the brewery floor. So that all worked really, really well, but that took us about 2 years to kind of get to the point of, okay, we can grow algae in the brewery floor and it's growing really well. And we know what kind of infrastructure we're going to need to make that process sort of work. I believe now they've got a bit more of a refined CO2 capturing mechanism in place, because what we had was a bit more of a sort of piloting exercise. So that's kind of all in place. And then it kind of had to tackle the question of, okay, so we're growing algae, we've got this other waste issue. How do we kind of combine these and maybe start to make impact with regards to methane emissions and existing supply chains that already sort of are in place with the spent grain? And that's where we went into a project with the Meat and Livestock Australia as well. And that project began in 2021. Kira came on board as the PhD student as that kind of project progressed. And that's been a multi-year project to kind of go, how do we take this algae, mix it with the spent grain from the brewery, test what the impact is on the ruminant methane emissions, and we got all the way to sheep trials. So obviously and I'm sure Kira will speak about this as well, very different project to kind of ones that implement macroalgae. Macroalgae does a bunch of different things with ruminants. But as I said, microalgae, there are so many different strains of microalgae out there, and we can kind of start to look at different microalgae that might do different things. Different microalgae put together can do different things. So really, for us, this project is still kind of underway, and I think there's a lot more to go in terms of finding more mixes that we can make and kind of getting to those next stages of trials with regards to cattle trials. I think, as I mentioned, one of the big things with this project, and I think one of the reasons why Kira, Rich, and I are so excited to speak today is obviously when we're trying to come up with these solutions, working with those solutions with people that are actually going to be end users is super, super important. And we've still got a long way to go with this project, and wanting to get insight and kind of feedback on how this can be more implementable is super important. Rich, I pinched this off your slide deck, but I kind of added some things to it because I kind of want to talk through where we've got to and where we're kind of going. So the timeline's kind of going, this is the projects we've done, but really what we've been able to do is kind of implement the brewery aspect of things. We can kind of capture that with microalgae, and we can kind of grow that into algae. And really, we have gotten a long way in terms of actually making that as a feed product, but we still have a way to go. And really the next stage with this is doing some more screening of microalgae, so we can really kind of find one that does what we want it to do, or find a couple that do what we want it to do, and then move to cattle trials with that, hopefully within the next couple of years. This stuff takes a lot of time, but as we've seen, I think when we consult more broadly with people that are utilizing these solutions, it becomes so much more implementable. And that's really the aim of this project, is to set something up with those supply chains, with end users, and obviously more broadly with Rich and the team, so that this can be something that's super viable in the future. Just a little recap of some of the people that we work with, lots of fun pictures and logos, some projects that I already mentioned. And that's really kind of my introduction, and I'm hoping now Kira is going to show some really cool data about that. What she's really going to talk about is this project here that we kind of have been doing over the last couple of years, in terms of getting in the lab, getting into those sheep trials, really kind of seeing what we've done, and obviously where we still need to go. So Kira, I think I'll hand over to you if that's all right.
Thanks, Alex. Just share my screen to make sure you guys are all with me before I get cracking. All right, everyone can see that? Perfect. So thanks for the introduction earlier, Paul. So yes, I am a PhD student from UTS. And I'm going to run you through essentially the last kind of 3 years of my life working on this project in collaboration with Young Henrys and the progression. So obviously we're here to talk about how we're going to use beer waste products. So in Australia, we love a beer, it's pretty obvious, but that means that we produce a lot of it. So we produce around 17 million hectolitres a year. And when we look at that in terms of our waste output, that's around 10,000 tonnes of carbon dioxide and around 350,000 tonnes of grain. So as Alex touched on, we came up with an innovative idea to solve the carbon problem with the use of microalgae. But we then had to figure out, what do we do with the microalgae once it's grown? So it's nutrient-rich biomass, so it's high in lipids, proteins, and carbohydrates. So they're all favourable components for use in livestock nutrition. So I came onto the project doing a PhD to look at how we can combine microalgae with the other waste stream, the brewer's spent grain, and distribute that as a nutrient-rich feed, with aims to reduce methane emissions, because we know that's an important topic that's being discussed, but also look at how microalgae inclusion affects productivity and meat quality in this context, because we're looking at lamb as well as beef. So I set out with 2 key objectives. The first being, how do we determine the effect of various species of microalgae and the dosage required to see our desired effect? So reducing methane without affecting other fermentation aspects. And then also looking at the key parts of animal productivity and meat quality. So determining the effect of microalgae we then selected from the lab work, to then use in a live trial to see how this affects performance, yield, and the taste and quality of the meat. So these objectives were then divided into 3 key phases. The first 2 phases were then conducted in the lab. So we had in vitro batch culture screening of microalgae. So as Alex mentioned, we have lots of options available with 70,000 predicted. But before we started this project, only about 30 had been screened for their effect on methane reduction. So we could see that there was a potential for microalgae to reduce methane, but we wanted to broaden that scale of how many species were screened. So we did a high throughput technique where we could screen lots of species at different concentrations to determine their effect over 24 hours. We then moved into a Rusitech system. So this system is an in vitro rumen simulation technique. So all that means is that it essentially mimics the cow's rumen in the lab. It's an open system, so this means that we can run a buffer through, have a closed fermenter, and run out the effluent to test the continuation of fermentation over a 15-day period. We run that 3 times and we can see the effect over a total of 45 days of the inclusion of microalgae on methane and fermentation. The third phase was then looking at the effect of microalgae in a performance pellet on lamb performance and meat and carcass characteristics. So for this, we had to move out of the lab and look at how this affected in real time in vivo lamb productivity. So I'm going to talk through phase 1 and 2. So the first step is we had to cultivate and grow the microalgae. So as Alex mentioned, we have a high capacity to do that at UTS, which allowed us to have a large variety of species to select from. So this would range from freshwater microalgae, which are typically high in protein, to marine, which are typically higher in fatty acids and lipids. Once we'd grown them, we then had to go out and collect rumen inoculum from our fish, not our fish later, but in collaboration with Sydney Uni, their fish lated cattle at Camden. We collect both the liquid and solid proportion directly from the rumen from a fistula, which is essentially like a window into the rumen. And we do this because the rumen microbiome is complex. There's lots of different things going on in there. So to get a representative example of a sample of what is going on, we collect it directly and bring it back to the lab. From here, we then conduct the first phase, so our batch culture assays. And so we use the liquid proportion of the rumen inoculum and we put that into different bottles, and these bottles contain different treatments. So say freshwater or marine microalgae at different dosage rates. And we run this for close them up, run it for 24 hours, and then we sample to see what was the effect of the microalgae inclusion on key fermentation parameters. So that would be methane, total gas production, and volatile fatty acids, which are a key energy supply to the rumen. And then we moved into phase 2. So from these batch screenings, we then could have a look at what algae species were having desirable effects, or if they're having negative, or if they're having none. And then we could kind of bring down that pool of candidates to 1 or 2 to then move into the rumen simulation experiment, where we then run one species at different dosages for that 15 days over 3 runs. And we can see how the rumen environment is being affected by the microalgae inclusion by looking at DNA. So we take samples from the liquid and solid proportion that we add in. And then we also can have a look at how fermentation is sustained over a longer duration. So some of the key findings from phase 1 and 2, we were able to screen up to 26 microalgae species in the lab. Most of these were novel and hadn't been screened before. At current, the reduction of methane is at 8%. So we'll be continuing at work at improving the efficiency of this reduction to our targeted 20 to 50%. So this phase, we learn a lot about the species and dose interactions and how these affect fermentation kinetics, and gave us a really good insight to what species were performing better and also our dosage range. So with high concentrations of microalgae, we saw a reduction in fermentation. And so this means it reduced gas and those volatile fatty acids. So then we were able to determine that a low rate of inclusion was most appropriate. From here, our 2 selected microalgae species that we used in the Rusitech experiment showed us that their inclusion didn't affect the microbiome, didn't reduce fermentation, but also that we didn't see a significant reduction in methane in these extended runs. So I'll chat at the end about what work we're planning on doing to improve that reduction efficiency. Phase 3 was then looking at the other objective of seeing what potential does microalgae have to improve, maintain lamb performance or affect meat quality. So microalgae are rich in proteins and polyunsaturated fatty acids. So there's additional benefits that they can have other than just focusing solely on methane reduction. So When we started this project, we understood we needed to take a comprehensive look at the whole picture. If we solely focus on methane production, we're going to miss a lot of really key other aspects. So this research aimed to investigate the effect of 2 different microalgae species, our fresh and our marine, and have a look at how it affected land performance, carcass characteristics, and meat quality. So to do this, we developed 3 different types of pellets in collaboration with Brewery Stock Feed, as well as Alex Chaves from the University of Sydney, who's the Professor of Animal Nutrition. And we included microalgae at 0%, which is our control, and then a fresh and a marine microalgae, which were our treatments. So then from here, we then had Dorper lambs as our test subjects. And so we had 3 treatments, 12 Dorper lambs per treatment. And these guys were fed for a diet of pellets and hay for 92 days on feed until the group's average weight was over 45 kilos, which we then slaughtered them and took samples post-mortem. So what we actually assessed in this experiment was performance parameters and looking at the effect of either microalgae species as a single effect. So what was the effect of fresh? What was the effect of marine? And then we also looked at microalgae dose. So what's the effect of no microalgae compared to including microalgae? And then key, we looked at the interaction between species and dose, which is considered our treatment effect. We looked at a variety of parameters, so it was quite a comprehensive study. So while we had lambs on ground, we looked at dry matter intake, average daily gain, and the feed conversion efficiency between these 2 parameters. And then post-mortem, we looked at carcass characteristics and meat quality. And so in post-mortem, we looked at cold carcass weight, hot carcass weight, initial pH. We then broke down these carcasses into and obtained 3 main cuts, so 2 from the leg and 1 from the loin. We then had a look at the colour. We then had a look at drip loss, so water retention, as well as pH. And then we then obtained the other loin to then run a sensory analysis trial to see what the effect of microalgae was on taste, as well as looking at the chemical attributes of the meat. We also then took it one step further. So we then wanted to look at what was the effect of microalgae on the rumen microbiome, because as you might know, it plays a really essential role in fermentation. So having an understanding of that level is really important to us. And then having a look at how microalgae affect the metabolomics. So we took plasma halfway through the trial and at the end. We also took liver samples post-mortem and rumen samples. And then we can have a look at the dynamics between the diet, nutrient utilization, and performance parameters. So let's get into the fun stuff, our results. So the key performance results, we saw similar dry matter intake across all treatments. So the inclusion of microalgae didn't reduce the intake. And so it was an average about 1.1 kilos of dry matter per day. So then it's no surprise that microalgae inclusion didn't affect our average daily gain or feed conversion efficiency. So this was kept consistent across all 3 treatments. And therefore we had a similar final live weight and cold carcass weight at the end of the trial. So there was no differences in weight gain. I also want to quickly chat about the fatty acid analysis that we did because we found quite interesting results here. As I mentioned, microalgae is a source of fats. Specifically, they are high in polyunsaturated fatty acids. So you might have heard of healthy omega-3s. That's what these fall into that category. And so we wanted to see if the inclusion of a supplement that was high in omega-3s could potentially affect the composition of the meat. So what we did, we collected liver, the subcutaneous fat we removed from the loin that we collected, and then we had the loin muscle. We then had a look at the fatty acid composition of all these 3 components. And what we saw with the liver to start with was that the inclusion of microalgae at 1% increased the concentration of these polyunsaturated fatty acids in the liver. And we saw a similar trend when we looked at the subcutaneous fat from the loin, that specifically omega-6 and omega-3 were increased. However, when we looked at the intramuscular fat within the loin muscle, we didn't see this effect. So what this is suggesting to us is that microalgae could have the potential to improve the omega-3s in edible fats derived from ruminants, which is quite an interesting result in terms of meat attributes. We then looked at how does it taste? So this is a big question that we found was getting asked when it comes to a feed additive that's fed to a ruminant that then we consume. So as you guys are probably aware, acceptability from consumers is crucial. So that is also influenced by taste, visual, and sensory attributes. So what we did we had a sensory trial with 42 participants. So this was set up in a triangle test. So essentially what we had were 3 pieces of meat on a plate. We asked them to eat all 3. And then we said, can you pick which one's different? So we had 2 from the same treatment and 1 from a different. And then we said, why can you pick the difference? And so that gave us an insight into why they had selected why that piece of meat was the odd one out. And so our key outcomes here was that there was no difference between the control and fresh algae fed lamb, except that we found that there was a perceivable difference from consumers between the control and the marine fed algae lamb. The key reason for this was that the control was identified to have a milder flavour and also be chewier than the marine algae fed lamb. So what are the next steps in terms of sensory analysis? So to understand consumer preference, we would then need to run a larger scale study to determine if our consumers would prefer the control or the marine fed algae. So that's also in future plans. So our key findings from stage 3. So through this lamb trial, we're able to see that the inclusion of either fresh or marine fed lamb did not affect animal performance, carcass characteristics, or meat quality. We were able to see an increase in polyunsaturated fatty acids, such as omega-3s in the lamb liver, as well as see that trend consistent in the subcutaneous fat that we removed from the outside of that loin. So where to next? With this project, as Alex has mentioned, it's still within its early phases. So what we're aiming to do next is to continue our in vitro screening, looking at a larger variety of novel microalgae species. At UTS, there's a high throughput capacity to run these experiments and also select ones that have optimal traits for what we're desiring. So there'll be continued research in trying to achieve that optimal methane reduction capacity from 8 to then 20%. As well as looking at synergy between the use of 2 species. So in certain cases, we see that using 2 microalgae species can be better than using 1 alone. They can have complementary effects. So now we have a bit more of an understanding about species and dose effect in the lab. We want to see if we can try to optimize this by combining 2 different microalgae species within the same diet. Once those objectives are achieved, progressing with in vivo cattle trials. So this is upscaling trials in cattle, having a look at metabolism, as well as in vivo methane production, because that's essential for validating our lab results. And then also progressing then to larger scale studies to make sure that this is applicable in an industry setting. And then another one Alex also touched on was supply chain logistics. So how do we get this out to you guys? But also how do we maintain a sustainable and reliable supply chain? So that's a key component as well. So that will be, now that we kind of have an idea of feeding ranges and dosage rates and production quantities, thanks to the early phases of this project, we can start to establish if a localized network would be better or if an interstate network would be better for this type of system. I also just want to say a quick thanks to everyone that's helped me along my PhD as I get the lovely opportunity to chat to you guys. It's not just me working on this. So we have a big research team at UTS as in terms of analytical and technical support. We've also had a lot of help from external collaborators, as well as the fundamental work the previous research investigator put into this in the early stages of the project. So that is all from me. If you have any questions, I'm excited to hear from you. And I just would like to say thank you for taking time out of most likely your busy day to jump on and listen to a project that we're really passionate about. So it means a lot if you've stepped away from the farm and the busy daily activities that are going on there. So thanks, everyone.
Great. Thanks, Kira, Alex and Richard, for what is a very amazing sort of project. So many insights and things that can be taken away. We'll now move on to our Q&A section of the webinar. So if you have any questions or would like to hear more about something, please feel free to put it in the chat or into the Q&A function. We've also had some questions that have been submitted online before the webinar. So we'll be pulling from both of those sources to answer. But we might start with one that's in the Q&A on the Zoom. And I'm not sure who's best placed to answer this, but just shout out, Kira, Richard, or Alex, if you think you know the answer. So was the 8% reduction, was that a result of actual methanogenesis inhibition or reflecting improved nutrient profile?
Yeah, so what we're seeing with that is likely due to an improved nutrition, improve the inclusion of fatty acids into the diet in terms of the polyunsaturated fatty acids. So unlike macroalgae, which we have the bromoform, which is a direct inhibitor, where so what we're seeing is likely that microalgae is an additive, and those fatty acids are resulting in a reduction in methane production. So it's not suspected that it's directly inhibiting methanogenesis. So we're now looking at ways that we can improve direct methanogenesis inhibition in our further studies.
Awesome. I don't have a science background, so I don't know exactly what methanogenesis and everything is, but I feel like that's a very technical question. So maybe another one is what is the intended scalability of these products? For example, large grazing herds, feedlot, small herds. Is there any insight that you guys have gathered throughout the project about those applications and where you see the scalability?
Yeah, we've certainly thought about it, and it's somewhat, there's a few ways we can tie it up. So we looked at whether you want to match the output of the spent grain in terms of a 1% feed additive, but we're looking at how most farmers are managing feed. That didn't seem to be the best solution. So it's probably more about sort of growing as much of the algae as we can based on CO2 that's available in the breweries, then just using that supply chain to get that there, and then having that managed as a separate application in terms of how getting the percentages right. We know with a lot of, particularly with feedlots, that they manage that that feed very, very closely, do monitoring of the micro biome of the herd. So rather than doing that work I think it's probably better to supply it separately. And then look, generally the hope is, and the aim is, that we get larger breweries involved so we can service more and more farmers and larger herds of cattle.
And then I suppose sort of along that same line of question, is there an estimated cost related to each animal depending on weight? What are the factors that contribute to the cost to a farmer?
Yeah, look, we're very conscious of what the market can bear here, and we understand that like from a feed lot, we can't be just piling on a whole bunch of extra costs without giving any extra benefit. I think at the moment, if we talk about sticks and carrots, there's probably not enough of either at the moment for farmers to really take on cattle feed supplement to reduce methanogenesis on its own. So there has to be a co-benefit there. And that's why I think, the findings that Kira talked about before around omega-3s and the flavor profile are really exciting. So if that's the benefit that we can deliver for a smaller uplift in feed, cost, and we understand the sensitivities around that. We can't go crazy about that, but if we can deliver that benefit there for a small incremental increase in feed costs, that's what we're aiming for.
And so I guess this question might also relate, because obviously the project's focused around methane reduction, but there's a question here saying, has there been any research into making feed for horses? So a market for domestic racehorse trotting racing areas, and whether that's like nutritional value for the horses, or has there been any research into that?
I'll take this one because I'm a horse girl at heart. So we've just started having a discussion about the potential of using microalgae in equine feed. So my understanding is that there's current interest in the ratio of omega-6 to omega-3. Exciting thing about microalgae with using co-species, we can look at satisfying that ratio, or using one species that contains the required ratio. So we're starting to looking at expanding our ideas around not just cattle feed, but is there benefits here for equine?
And so I guess as well, so there's another question, that's what does success look like? So I'm guessing at the start of the project, it might have only been focused around a few key areas, and it seems like it's sort of grown. So what does success look like for you guys with this project?
Look, we've probably got different targets here, but I'm looking at it from a real business opportunity here to say, you know, is there a complementary industry that what sits alongside brewing operations, and then more broader, I think, and a couple of smart people in the Q&A have picked this up. Is it like, okay, well, what are the other CO2 emitting industries that we can tap into here? Because it really is about a source of CO2, and then looking at what makes sense from a circularity perspective in terms of co-locating industries. So there's a lot of opportunities out there. We're super excited to be kind of in the forefront of this, and then working with experts of UTS to say, hey, what are the solutions that we could come up with and potentially create a whole new industry.
And so then there's another question. Well, I'll let Kira.
Sorry. Yeah, because I have a different answer, Richard, so I thought I'd be an artist's perspective. For me, success for this project is collaboration between farmers to develop something that is useful for them. So developing something that we can have a reliable supply chain that isn't hard to feed out on the farm, that doesn't implement them to a great degree, but also has benefits in terms of that sustainability aspects of methane, but also not affecting productivity or meat quality, but more importantly, trying to enhance those aspects for them in a combined manner.
Great success targets as well for both of you guys. So I guess sort of another question here is about whether you envisage this becoming an industry standard in brewing, and having other brewers and maybe large scale breweries around the world sort of taking this on. And is that perhaps a reality one day that you see?
Yeah, we've certainly had a lot of interest from breweries around the world. People are interested in tackling this problem, and obviously, if we can turn what is to waste streams into revenue, that's of interest to everybody that you're talking to, right? So, yeah, that really is the aim. We don't want to keep this under the wraps of Young Henrys, and if we are going to have a global impact, we really need to spread it out. So that is the aim for sure.
And then I guess as well, I think this question here is aimed at for you guys. You know, is there a sense of the cost increase per ton of spent grain to apply the tech at scale?
Yeah, there will have to be, right? So that's what I'm saying. We need to identify that co-benefit, and then what the market will bear in terms of the cost of that co-benefit, and then the reduction of methane genesis is almost just the the icing on top. But really, that's been our main game. But we understand that the way that things are now is, as I said, that neither the stick nor the carrot for farmers to implement methane reducing technology exists out. So there has to be another benefit alongside it.
Yeah. Maybe looking at some of the questions that we got beforehand online. So is there, I know you touched on this, Kira and Alex, about, the health effect on the trial animals. Was there an increase in sort of the health outcomes for those animals in the trial?
So in terms of health, what the parameters that we looked at was the productivity. So if they were maintaining productivity to that of the control, ideally we would like to see that they had an increased growth rate, but that's not what we saw. But we didn't see a decrease. So we can say that they were as healthy as our control animals that didn't have the microalgae inclusion. On a deeper level, we also looked at the metabolomics, which can give an indication of there what's happening kind of under the surface, and we saw no differences between the control and our treatment animals. So yeah, we can say that it didn't negatively impact their health from what we observed in the study. But the term of health, we can definitely look into that, looking at specific indicators in like blood, plasma and things, and get more refined there once we move into cattle trials as well.
Right. Someone's also put in here, next pub meal, Young Henry's beer and Young Henry's 3 improved omega-3 improved steak. So sounds like there's plans in the works for this down in the future for people going to the pub.
Yeah, we can do like a tasting of the two.
Yeah.
I've been trying to answer as many questions as possible as they pop up. I think that's the teacher in me as well, having to manage multiple lines. I did see a question from P. Smith saying the supplement accepted as livestock in terms of taste. Yeah, that was a great one of the great findings, Kira, wasn't it? From the lamb trial.
When I reference dry matter intake, so that's just like in reference to palatability and how they're eating it. So we didn't see any difference when we specifically looked at both total dry matter intake as well as pellet intake. So we collected refusals of hay and pellets separately, so we can make sure that we were actually looking at the pellet intake, which included the algae in it, and that was consistent with the control. So we can say that we didn't see any reduction there.
Right.
The other one, Kira, was, do you know of any microalgae producing anti-methagenic substances? So I guess that's maybe the bromoform. Is that what I guess maybe we're not aware of microalgae doing bromoform, and we've gone down a different angle. So essentially,
so microalgae a little bit different to macro. So your red asparagus sits in that macro category and is the bromoform containing compound. We haven't specifically focused on bromoform just because we wanted to take a different approach with using microalgae and seeing if there's other alternative compounds. So that will be the next phase of this project, really honing in on what compounds are having a specific effect on metagenesis. There are a few that I'm aware of. So yeah, there's a couple there that we'll be looking into, but I'm not sure how much Richard wants me to say about the specific compound. Yeah, otherwise I might. But yeah, there are one or two, but they're different from the one that's quite commonly spoken about, which is the bromoform.
There's a question here about sort of grazing enterprises and daily pellets. I know some other sort of methane inhibitors have looked at lick blocks and the other sort. Is there any sort of thoughts about down the line how to incorporate the project and the product into grazing enterprises?
Yeah, so I think that in that conversation, there's two key technologies that are being produced. So you've got your lick block, so sit in the paddock, opportunistic, come up, have a lick, and then they walk away. But then also the development of kind of like a mineral bolus technology, where not in that phase of them moving into that over the next few years, but that's something that we'll have to look at diversifying into just for inclusivity of the industry and trying to get out of just providing something for, say, feedlot setting. So yeah, they both will have their challenges in mainly with the lick block dosage requirements. So having a look at how we could kind of manage that in that scenario. And then also bolus technology, there's the processing side. Once you head to the abattoir, making sure that we're not interfering in that side of the supply chain as well will be an important consideration.
There's another question as well about home brewing and whether there's a way for home brewers to be involved and use and provide their spent product. I don't know if you guys have thought about that. No.
Not at this stage. Yeah, look, I remember being a former home brewer. We are talking about tons in this stage, so scale is certainly part of it. Look, I don't know. I'll see if I can think of something in terms of making that work.
Yeah. And then I guess as well, there's a few questions about marketing. So, how do you market this product?
Yeah, we've certainly got a lot of ideas around that, and with along with Beer Positive, we have Beef Positive as a trademark. So, looking at a form of certification may be a pathway we go down. Look, the big retailers are always going to be grabbing any chance to spruik a benefit of their products. So I'm not particularly worried about that at this stage. I think if we can do the hard work on the science and the supply chain and make it work, and we can show a benefit, both in methane reduction and, hopefully on the omega-3 and the flavor profiles as well. That then, it's something that's going to sell itself in terms of marketing. I'm massively concerned, but really, yeah, the marketing benefit to breweries along with the financial are obvious, I think, as well, to say that you're part of a movement here to have circularity amongst your supply chain and the agricultural sector is a fantastic opportunity to get people excited about drinking your beer.
Right. Yeah, there's definitely a lot of opportunities there, from what I can see. There's one final question sort of that's popped up around using spent grain as a mushroom substrate, or as an ingredient, as a potential food ingredient. I don't know if you guys have thought about that or know any knowledge. It seems like your project is largely concerned around the rumen and how
Yeah, I know
that works.
you can grow mushrooms on it. That's if there's mushroom farmers out there, yeah, it's not a bad feedstock for that as well. And in terms of using that, it's very rich in protein. I know some bakers have used spent grain to produce bread. Just in pet food, another person mentioned pet food. I know people have made like their little doggy treats from spent grain as well. So there's a bunch of different uses for it as well.
Yeah.
Yeah.
And there's another one that, have you considered delivery in stock water? So delivery of the pellets in water, is that something you guys have looked at or thought about?
In terms of, so kind of take that into two aspects. So in terms of storage life and delivery, we then have been speaking about a way of drying the grain to improve its storage life, and then as well as with the algae, because if we produce a wet product, it's going to be affected in that storage component. In terms of pelletization, I guess our biggest consideration is then, do we directly supply onto farm the two components separate, or do we supply it to a pelletizer that then combines it into a pellet form? And then there's also been discussion about like a liquid supplement that could be like a mineral liquid supplement, and the potential of supplying algae in that form into the brewer spent grain. So as we said, still early, there's lots of options that we're now seeing arise where this could fall into, and knowing what we know now. So hopefully that clarifies that question.
Right. Well, I think we're probably approaching time, and I'm not seeing a lot of other questions come through that we can answer sort of live. So we might wrap things up. But thank you so much, Alex, Kira, and Richard, for joining us today. You guys are doing amazing work, and it's certainly a great project, especially thinking about the circular economy aspects and everything else, and be a positive. What a great catchphrase. So we'll wrap it up there, and just say, if anyone has any questions down the line, please feel free to reach out to us at FCA, or to Richard and the team at UTS. I think they had some of their contact details as well on the screen. I'll say as well that the recording will be made live on YouTube afterwards. So we'll post that up on the internet, and also send it out to everyone who signed up via email. If you're enjoying FCA's webinars, this is the first one we've had this year. Keep an eye out for our upcoming ones. We have a lot in the work for this year. And then there's some ways there that you can get involved. So we'll close it out there. So thank you so much, Kira, Richard, and Alex again. And have a great rest of your afternoon, everyone.
Thanks, Paul. Thanks for attending.